Okay. Hi, everybody. Thanks so much for coming today. I really appreciate it. I'm always delighted to be able to come to these talks and talk about some of the research that we've been doing. Most people that know me as like that lawyer guy who has a contracting company. So it's such a treat to be able to talk about the work that really has been inspired Spadefoot Design and Construction, and now it's newly rebranded as Spadefoot Ecosystem Solutions. I really appreciate it again for the opportunity to talk about the work that we're doing. We Spadefoot spent the entire winter, all of our technical staff stayed on and, really pushing forward this research agenda that has undergirded our entire work that we've been doing and really our future directions. A background on the theory. I wanted to first go through some of the work that we've been doing, the work I've been working on for 20 years, and I'm happy to have like almost a full-fledged lab at this point supporting the work. There's five of us plugging away all winter. So it started sort of humbly, right? I said I'm a lawyer. I have this contracting company. However, I started as a aspiring herpetologist. For those of you who don't know, herpetology is the study of reptiles and amphibians. And I had a very seemingly simple research question. Basically, why are the salamanders where they are? And you can see a picture of a spotted salamander that I found at West Hills County Park. That was one of the focal organisms that I studied. It was a very interesting journey for me on this research paradigm because the first thing I encountered was discomfort from my committee. I wanted to figure out why the salamanders were where they were, and they wanted to know what experimental manipulations I was going to be doing and so on. And so I actually did what it amounted to two theses, at least collecting the data for two theses. I did all this morphometric data that did not appear in my thesis at all because my advisor, probably rightfully so, wanted to make sure that if I went through the whole program, at the end of it, I actually had enough data to write a thesis about. So what we did is we went out and put a drift fence array. It's a sampling technique for salamanders and herpetofauna, also insects as well. Essentially, all it was silt fence dug into the ground, and we dug buckets flush to the ground. And so what happens is that when these salamanders are migrating, I should say, to their breeding pool, they walk along, and they're going straight to their breeding pool from the terrestrial habitat. They walk into the silt fence, and instead of trying to go over it, they go along it and fall in the bucket. So, and then every single day I went out there, pulled the salamanders out of the bucket, put them on their other side, counted them, measured them, and I had a pretty good, abundance measure. I quickly realized that I couldn't figure out why the salamanders were where they were without understanding the patterns of vegetation. And, so what started as an interest in salamanders quickly became an obsession with plant communities and, why the plants are causing the salamanders to be where they are. And I have a note here about controlled studies in nature. My viewpoint is that it's largely impossible to do that. There are some exceptions. Obviously, if you set up a good controlled experiment where you're treating one side and not the other side, yeah, of course, that's a pretty good, it's a pretty good setup. However, even in those strict circumstances, you're not going to get it. There's always going to be conflating factors, climate, sun, shade, soil texture, mycorrhizal communities and stuff. So I set forth to come up with a research paradigm that can actually just basically measure observationally what's in place and understand why things fit together. Funny enough, I started taking this data before I even knew how to analyze the data, which is kind of to show you my mindset here. So what I came up with over time was something called was a distinct application of fuzzy set theory. Just to back up a little bit, classical set notation assumes that you're in or out of the category. You're in the wetland or you're out of the wetland. Fuzzy sets, however, allow for and mathematically allow for that continuum. Everybody knows, or anybody that's been outside understands that, if you're in the water, you're probably in the wetland. And if you're dry, you're probably in the upland. But there's a whole gradation in between where it's not so clear which category. It's really partial membership of the wetland set. And so what we did to make things comparable, to make tree basal area, leaf litter depth, burrow density, and salamander abundance to be able to be compared mathematically, we first had to fix the fact that they're all measured on different scales. So we do something called min-max normalization. So we basically key everything between 0 and 1. And so this way, salamander abundance is directly on the scale with pH, with canopy cover, with leaf litter, whatever it is that the variables that you're measuring. And this way, they're able to be they're directly comparable. And so we use ordination, typically principal components analysis, to kind of flush out which what are the major gradients of variation? So, we described that wetland to upland variation gradient. Think about, though, in nature, there's also layered on top of that. There's soil texture gradients. There's canopy, cover gradients, shady to sunny areas. And so these are all multiple interacting gradients that fuzzy sets allow you to kind of take apart and compare the relative influence of each. And so what we tried to figure out is what is the partial membership across these respective gradients. Right now, we have two papers under peer review. I've been kicking around with the editorial office with the first one, the salamander paper, for months now. I just submitted a second one, which is really more of a policy application of fuzzy sets. It's a short one. It's a short demonstration paper. But basically, the whole premise of it is that overlap is inherent to nature. The boundaries that we're drawing are just political decisions. And if we can separate that, the science from the policies of line drawing, I think that the science and the policies of line drawing will be doing much better. So those papers are under peer review. So we went from salamander habitat to ecological fields, essentially, is what we're measuring, modeling, and comparing across time and space. And so what started with, hey, why are the salamanders where they are, went to our ecological fields scale invariant. So I actually had the pleasure, I see Mina sitting right there. I had the pleasure, it was the first public comments I made about fuzzy sets in, 15 years or so at the Cornell Cooperative meeting. And it was an honor to do it. At the time, we just had one data set. It was at a 2.5 acre scale at West Hills County Park. And we spent a ton of time on that presentation going into very, the minutia of the operational details. But I said, hey, I think we're going to try to do this at different scales. It should work. If my theory is right, it should work across scales. And so we spent a boatload of time out in the woods at West Hills County Park and collecting other data sets and basically running it through the workflow. And we tried to break this model over and over again. And no matter what we do, we keep getting the same scale invariant structure. So you can see here, 2.5 acres, 100 acres. We used Whitaker's canonical data set from the Great Smoky Mountains. He's the old school gradient ecologist. He's the guy who brought the concept of gradients to the field of ecology. We took his data set from the 1950s. It all organized out the same way. We have some awesome data from Curacao. We did coral reefs. The same things happened. 18.6 million acres of data from the forest inventory and analysis, ran it through the workflow. The same rules popped out. Temporal pond monitoring data, wastewater collection systems, human social systems, and Yankees baseball. Any system, dynamic system under constraint seems to have the same thing. And I have a question mark here with black holes. I recognize in advance like how crazy the slide makes me look up here, but at the same time, this keeps working, and I'm going to take you through 3 scales today. I'm going to try to do it simply and to show you that I think, and it's not a universal law. I don't know when it gets there, but I'll tell you that any system, a dynamic system with energy inputs under constraints, their same orthogonal roles start to pop out over time. So what are these roles that continues to appear? You see Nick, one of our field technicians at Spadefoot, doing sludge judge monitoring, sludge depth monitoring at Sands Point Preserve, where we're doing a bioaugmentation study. But pretty much always the PC1 is basically what is the overall state space. So in the context of forest, that means, what is the vegetative biomass? What is the structural components of a coral reef? How thick and functional is a biofilm in a sewer collection system? It's almost always this overall state space is PC1. I said almost. Actually, every time that we've run this, it's always the overall state space on PC1. PC2 is an alternative stable state. In nature, anybody who's familiar with some of the ecological literature, they've heard this term before, alternative stable state, or the term intermediate disturbance hypothesis. So basically, if you have a forest and you introduce some, as an example, and you introduce some water to it, it creates another stable organization, a forested wetland. Forested wetland species, a lot of them, such as some sycamores and box elder maple, they typically do not make it to the canopy unless there's modest disturbance associated with ongoing stormwater. In the context of coral reefs, of course, it was the exposure to the ocean. And PC3 is always in every one of these workflows that we've done over the years has been drift perturbations. So for coral reefs, that is nitrogen dynamics, so nitrogen overload. For forests, it's invasive species proliferation and edge effect. So these, I'm not saying that coral reefs, wastewater systems, ponds, and forests are the same, but I think that they're organized similarly. And underneath all of that are redox gradients. There's more axes of interest, but let's not go too far down the rabbit hole. I think that's probably enough. So here's some actual data here, because for now, until up to this point, it was just me kind of opining that these roles are similar. But if you look at the on my right, I think it's your right too. Yeah, your right too. It's the eigenvalue decay across spatial extents, and they're remarkably similar. The orange one is 18.6 million acres worth of data from the forest inventory analysis, something like 3,000 plots. The coral data sets are on here. West Hills and Janes Hill are the two scales that we did, and I'll take you through in a moment. So their decay rate across eigenvalues are remarkably similar. And what you look on the left, you look at this sort of the correlation heat map. You see some of these R values that are above 0.9, 0.96. So coral data set from 2015 and the Great Smoky Mountains data that Robert Whitaker took in the 50s are essentially equal from their eigenvalue decay, which is pretty remarkable. But this is just geometry, right? You could just say, oh, maybe this is just an artifact of the mathematics that you're doing, right? It's not, because what's below these artifacts are very explicit mechanistic details showing analogous roles appearing across these different dynamic systems. So I'm going to try to take you very quickly through three scales that we did. The 2.5 acre scale at West Hills County Park that I had the pleasure to speak at CCE event last year, 100 acre scale in the same park. You can actually see right here on the diagram. This is Janes Hill site. It's about, I believe it's 30 plots, if I'm not mistaken, across about 100 acres. And then we did about 14 plots across 2.5 acres. So they're right next to each other, but they're different data sets. And then we're going to go through data that we culled from the FIA data. Back in, I want to say, the 30s or so, Congress mandated that the U.S. Forest Service keep track of all of our forests. It's good, and so they have a grid system out there where they're, taking all this awesome data, mostly about tree data, from all across the nation. We pulled down all the data, and it was a massive, massive data set that we downloaded and processed. But we pulled down the data set from all 18.6 million acres of New York State, and I wanted to see if that broke the model. It didn't. Spoiler alert. But I also want to talk about, as an example of some of the mechanistic details that are built in, I want to talk a little bit about how American beech is changing across scales and how, it's sort of relevant to this discussion. So these are what the ordination diagrams look like. In the past, I've at talks. I mean, gosh, I can't even see it from here, to be honest. Let alone the folks in the field. So there is a mechanistic detail built into these. Every one of these vectors and points here makes sense from the natural history of the species at reference that we're referencing. But what we do often is we'll take each individual axis and project it into actual map space, the spatial extent, so that we can understand how these different gradients propagate spatially across scales. So this is PC1. for West Hills County Park, that smaller 2.5 acre data set. So represented in blue, it's what Robert Whitaker referred to as a complex elevation gradient. So basically, to kind of summarize, the blue areas are higher elevation. They're more dominated by oak and ericaceous species, and so those are the blueberries, the mountain laurels, species that have associations with mycorrhizal fungi. You see the notes here, ECM, that's ectomycorrhizal, and ERM, ericoid mycorrhizal fungi. Those are the ones that are associated with blueberries and laurels and whatnot. And so the areas that are in the blue areas are, more drier uplands and have slower decomposition and more conserved nutrient cycling. And so when we look at American beech here, it's weakly correlated with the forest. And what that really meant was it's not explaining a whole lot of variance at all within the data set. And I think that makes sense because this entire study site, this 2.5 acres, 14 plots at West Hills County Park, was all on the northeastern facing aspect of a hillside. So it's all within the realized niche of an American beech. So it wouldn't explain a lot of variance. But I think the key thing to look at on this map, high elevation areas are in the blue and low elevation areas are in the red. The topography we added after the fact, keep in mind this image is just generated from vegetative patterns in the forest. So we didn't feed any elevation data into our models, into the spatial projection. It's just what's popping out based on the vegetative patterns. So I got to present this at CCE, kept mentioning that because it's the first talk that I gave. And I was scratching my head wondering, gosh, if this is true, it should be at every scale, right? And here we are. We did another sample, and you can see the elevation here. This is the high elevation spot, and these are the lower elevation spots. Again, we didn't measure we didn't model any of the elevation data. All these patterns that you're seeing are derived solely from the vegetative community. And again, I'm not going to repeat the life history and sort of the nuance about what makes a PCA1, but I did want to point out what happens with the American beech in this Janes Hill model. What's fascinating about the Janes Hill model, I think, is that there's different aspects in elevation. So it's a very diverse topographic area. So we have the contrast of south-facing aspects, which are not like the happy zone of the American beech. And when that contrast became evident, beech pulled into a major driver of the variation in the data set. So we actually named the axis on that scale after the American beech as the stable music set, kind of adopting some of the language from Robert Whitaker's canonical study. And what we've noticed now that context is evident is what the red maples and the tupelos are doing. So all of the seedlings and the saplings, for the most part, are associated with the stable music, beech-dominated northeastern sort of hillside kind of community in West Hills County Park. But the adults jumped way to the other side, and we're about to talk about the AM set, that alternative stable state. So what it appears based on our models is that American beeches are really a dominant driver of community, assembly, and the tupelos and the red maples, are very happy to live in the understory, and they do it in two different ways. The red maple seedlings, they seed out like crazy, and tupelos tend to have like oaks. They'll tend to live and live for a very long time under the canopy. And so when things get a little bit too wet or the beech trees die back because of, beech leaf disease, for example, you start to see the tupelos and the red maples that were existing under these beech canopies absolutely exploding into the scene. So, we can go through the ordination diagrams. I have them on the back, but I'm trying to summarize, years of work here. And here we are in New York State. And you can see, again, this elevation. Anybody who knows some of the geography of New York State, this is the Adirondack Mountains. This is the Catskills right here. This is the Allegheny Plateau, higher elevations points in New York State. And again, I just wanted to point out that we never measured or fed elevation data into any of these models. All we're doing is modeling the co-occurrence across partial memberships, for plant species primarily. And I'll say here, the American beech is still the driver of the stable music set. So, this is these red spots are kind of beach happy zones. Okay, PC2. I'll be quicker because I think you get the idea about the scale and variance a little bit. But this is essentially lower slope positions. Adult maples and other arbuscular mycorrhizal species. So these are things like some of the grasses, some of the white wood asters, jump seeds. These herbaceous plants are much more prevalent in this alternative state. So there's much faster organic turnover, and there's intermediate disturbance. Again, these species are happy there because of some disturbance, usually associated with shallow subsurface flows and the expression of the aquifer and the groundwater kind of moving through as sheet flow. Here it is in Janes Hill, same thing. Again, these are completely separate data sets, different observers actually, and we're just feeding in vegetative data, and you can just see the lower elevation areas are in red, and the higher elevation areas are in blue. Here it is in New York State. This one gets a little bit harder to see, but you can see the high elevation areas are in the yellowish kind of range. And the lower and sort of blue tints, especially in the Adirondack Mountains. But you look in the Finger Lakes region, Long Island, the Hudson Valley, that's, that same alternative stable state. It's popping out in the vegetative data on a state level. And this one is, and I think maybe most relevant to LIISMA and the work that we're doing here today is the disturbance gradient. So this is on the small level, it's really what most naturalists would refer to as edge effect. So you can kind of just see there's a beautiful forest, but adjacent to the forest, I mean the human development is just basically bleeding into the forest in the form of burning bush and garlic mustard and English ivy and all these nasty invasive species that we're continuing to fight with. So in these circumstances, there's a rapid organic turnover. Most of the mycorrhizal associations are generalist arbuscular, so really primitive mycorrhizal associations, or non-mycorrhizal species. So Japanese knotweed, tree of heaven, these species, they actually go after the mycorrhizal community via allelochemicals, that the roots put out. So what's happening with plot 5 though? Because plot 5, with the going theory, it's not really near the edge, but there's still clearly some degree of disturbance on PC3, or at least that's what the model would suggest. Sure enough, we went out there and field ground truth that there must have been some sort of like microburst or something over time. So that plot was up on the ridge, and there was a lot of down trees, and the mountain laurels was a little bit more robust there, but perhaps there was some, gypsy moth defoliation or something, but there was definitely disturbance in down trees in this area, which is interesting. But it wasn't as bad as just, against the road here. Going up to Janes Hill here, you can see that there's a road. I probably should put the roads on this, but there's a road right here. And the nastiest spot is, adjacent to the water tower and some human development. It's really just, again, this edge effect. So it's very interesting that this kind of popped out, again, just from vegetative species that we're doing. Okay, let's get into the practical applied part. About halfway through, I see on my time, so that's good. So the research implications. First, none of this is mystical. We do know why the salamanders are where they are, but nothing that I said was anything today, kind of describing these models. I think it's remarkable that the math basically naively put these patterns together for us. But what the patterns mean is just basic field biology. Anybody who's been outside and been thoughtful about gradients in space, it's not so mystical, and so we are doing some good work with this. We're trying to put together resources and not make, we're working on papers, but more importantly, we worked our tails off this winter on something that we're going to refer to as the Natural History Field Atlas. So if anybody is familiar with USDA Silvix, it's a wonderful resource from the old school foresters, and they kind of put together what trees are doing what over time, and it's really like a critical resource. So what we're doing is sort of updating it for 2026 and tying it. So the Silvix was I think accused unfairly of being a little bit more descriptional, observational, and so on. We're tying our species accounts to actual field coordinates across different sampling scales. And so basically what you'll be able to do on our field atlas for any of the species that we've encountered, and this is not just for the forest, we're doing it for the coral reefs, we're doing it for, whatever new data sets, we're just going to have this ongoing manual. The three gradients, I'm sorry, four, if you count the Whitaker stuff, the entries now span 500 pages, and that's what we wrote all winter. And so we're right now digitizing this so that you can actually query by species name or go into one of our ordination diagrams and like literally click on the variable, and it'll bring you to the species account. And what you get out of it is, it'll be some interesting stuff being tied to field coordinates, but you'll also be able to understand, okay, what does this plant do in nature based on the research that we've done? So, what is its shade tolerance? What is its community type? What mycorrhizal associations does it have? And what evidence do we have that this is not, based on our actual quantitative data and our mappings, like what evidence is it that, we're making these premises on? So we're really delighted to be able to offer this free open access to anybody. And we're also planning on putting the workflow online so that if you all have these incredible data sets that I know you have, you could just, at some point soon, you could just go onto our website, upload an Excel file, press execute, and it'll just spit out all the ordination diagrams, all the spatial interpolation. So, I think that this will be more powerful if it's adopted. And so we're doing our best to make it a resource for everybody who's interested in using this resource. And so we're able to use, I think the most powerful application of this is adaptive management. And so to give an example, we're doing some bioaugmentation studies here on Long Island with a technology that Marshall and I brought to the island. And what we're showing is that the state space is actually moving over time. So variables that started high on the drift set, that disturbance perturbation set, but low on the overall state space set, are drifting towards the centroid as things, towards the middle, and reducing their membership in the disturbance gradient and increasing their membership on this overall state space stuff. So we're able to actually monitor in real time how these overlapping gradients are changing over time based on interventions that we're doing. So I think this will be the most powerful application of this work. And the overall theme is, and this is sort of, I think, kind of what LIISMA is, although framed very differently, is reducing ecological drift. So again, these are not mystical things. It's, what are we doing to make nature better? Yes, we can monitor it with fuzzy sets, but we all know the recipe already, right? It's invasive species removal, it's native plant community restoration, and it's stormwater management. We do these three things well across Long Island and beyond, and we'll be in good shape. We continue to do the other way, where native plant communities and stormwater management and invasive species are not really considered during, let's just say land use hearings, for example, we're going to be in trouble, right? So what we're doing is Spadefoot Ecosystem Solutions is operationalizing the theory. So what is it? What are we doing? Simply what you folks are doing, and quite wonderfully, I'll say, is removing invasive species, planting native plant communities, and managing our stormwater. So there's a clear connection between the prevalence of invasive species and aquifer protection and surface waters protection. So you'll see on the right, this is a beautiful red maple getting strangled by wisteria at West Hills County Park. You could be happy the red maple has a new lease on life. We got rid of that wisteria vine. It met its end with our excavators. But at the same time, you could think about this picture every time somebody says, "Oh, it's not invasive in my yard." It clearly is. And on the left, you have what I think of as a big giant dandelion, which is the Norway maple. And if this was an oak, you couldn't really I couldn't put my arms around how big this Norway maple is. If it was an oak or even a native maple, the root structure would be twice as big as the drip line of the canopy for the most part. This root ball was basically nothing. It was basically the trunk of the tree. And so when we have these aggressive invasive species, destroying our native infrastructure and, being poorly rooted, crowding up, growing up. You have the deep-rooted natives that make these incredible, important connections with the mycorrhizal fungi are not allowed to proliferate. So I think a lot of people think, oh, let's control our wastewater and then we're in good shape. I've never suggested that we shouldn't be controlling our wastewater. However, the whole processing mechanism of the ecosystem is the natural areas around it. So just dealing with the wastewater is not going to save us from all these brown tides that we're experiencing. It just won't. We really need to restore our ecosystems. And I have a note here about invasive species actively working against mycorrhizal networks. I should probably go back, and I have some time here to talk, what are mycorrhizal fungi anyway? So the key adaptation that plants made when they left let's call it the primordial soup and made it on land, was developing a symbiosis, or more specifically a mutualism with mycorrhizal fungi. And so these fungi, it's this incredible nutrient economy that is existing all around us that a few people, I should say, think about on a daily basis. I'm one of those weirdos who do think about them on a daily basis. But at the same time, it's incredible to think about. So what happens is the mycorrhizal fungi go into the soil, they find nutrients, and they bring them to the tree roots. And the trees, in exchange for these nutrients, the trees give them carbohydrates, sugars from photosynthesis. It's an incredible adaptation, and the fact that the forests as we know it, and meadows and any other natural area, they owe their dominance and their beauty and their structure to the symbiosis with mycorrhizal fungi. And invasive species are doing chemical warfare to kind of kill the mycorrhizal fungi. Believe it or not, the invasive species that I hate the most is garlic mustard, and it's for two reasons. One, we do a really good job of getting rid of the invasive species. The burning bush is gone, the English ivy is gone, the vinca is gone, and then you come back and you're treated to, hello, thank you for freeing me up from the light, and you have this whole carpet of garlic mustard. So it's a pain in the tush because we always have this secondary invasion, or a lot of times you have the secondary invasion of garlic mustard. But the other thing too that it does, and this is pretty insidious if you think about it, it exudes chemicals that kill mycorrhizal fungi. So if they are in a situation where the common mycorrhizal network, as it's referred to, is healthy, they can't really do well and proliferate. The same thing goes with knotweed, by the way, and tree of heaven. You ever notice that knotweed basically stops at the forest edge? It's because of the intact mycorrhizal network. So when we're pulling out these invasive species, especially these stinking little garlic mustard seedlings that are just going to seed right now, you're actually reducing the amount of chemical warfare against the mycorrhizal network. And that's why after a couple of years of pulling and a couple of years of management, I'll say the garden is never done, but it gets a lot better after 3 years. And that's essentially contemporaneous with when the mycorrhizal networks develop. So native plant community installation. So one of the applications of fuzzy sets and some of the, not just the explicit measuring and monitoring, but also some of the more observational work that we've been doing over the years is trying to figure out when you're planting native plants, when do you even need to plant them? When is taking out the invasive species enough? And so it's really not rocket science. I guess it's plant ecology, I guess. But really, a self-heal is likely to be successful in places where there's fewer invasive species and more native species, right? This is to file that under duh, right? But at the same time, there's this push towards, hey, we're going to let nature come back on her own. Nature's tough, but sometimes we need to push in her directions. So this plot right here at the Science Museum of Long Island, that was about an acre of kudzu. Believe it or not, on Long Island that we took out mechanically. And now there's, for the most part, it's just beautiful native forest species that are there. The only exception, and you can kind of see it around here, there's an old concrete like foundation or whatever. And we've been grateful for the partnership of the DEC who's been coming in and spraying herbicides in a spot, that we couldn't get to mechanically. But we took out an acre of kudzu and it stayed out. And it wasn't because it was scared of us. It was because the native plant communities knit together and really retarded the invasive species coming back. I wanted to also talk about when we're designing a restoration project and a replanting, we try to think about the community over the individual plants. And so what I mean by that is that whenever we do a planting, we're planting early successional species that'll grow up quick, start juicing up this mycorrhizal community, and later successional species that we know are going to shade out and kill the early successional species over time. And it could be as soon as, 15 years from now, or even less, depending on which species they're talking about and the proximity that we're planting them. But this is an important consideration. When we're doing these restoration projects, survival rate is important, but being able to understand the trajectory of the system over time is more important, I would say. And so areas that are really mucked up with an invasive species, we need to plant densely, and we need to plan for ecological succession while we're designing our planting community plans. And that was my last bullet point there. And managing stormwater. And so I mentioned that it's a really interesting dynamic. PC2 creates this alternative stable state. It's really just some stormwater. It does well because that's this braided upland wetland community that we really try to push our systems towards. And so our group at Spadefoot, they've actually like taken on this parlance. We're trying to make it more PC2 than PC3. And so we're hoping that this catches on. Probably won't. That's okay. But the point is that the state space tracks this over time. And so one of the things we're trying to do with our work at Spadefoot is try to take advantage of the power of native plants in order to actually do stormwater management. So most land use hearing, when we're talking about stormwater management plans, these SWIPs and whatnot, they think about it in terms of storage. Where are we going to put the water? And so everything's a dry well, everything's a French drain. It doesn't really matter if there's like a clay layer 2 feet underground. We're still going to drop a dry well in and pretend it works. And so the work that we do is that we try to take advantage of above ground storage, this sort of freeboard that you get when you have these like bioswales and rain gardens. And in a lot of situations, plants are better at managing stormwater as long as you're carefully designing for the overflow of these rain gardens and whatnot. The plants are better than precast in managing stormwater. And that's because, the day you install built infrastructure, it's at its peak efficacy and it degrades in its efficacy over time. The day you install a native plant community, it'll work, but it only gets better over time, especially if you fight off the invasives that are inevitable for the first couple of years. And I'll note that trees as stormwater powerhouses. You could think of every tree, especially the native trees, as being sort of a straw on the ground. And so collectively, the wisdom, if you want to call it that, of Long Islanders is every time they move into their house, the first thing that people do is cut down all the trees. They don't quite get it, and then they wonder why all this water piles up and Stony Brook is blowing out and so on. It's really I think blamed in my opinion on the fact that we're taking out all the trees and canopy structure throughout Long Island, and so plant them back, please. When I grew up in North Massapequa, I remember my entire block was lined with oaks. There was a canopy. It was cool. You go down there now, it's like sort of an ecological hellscape. I'm sorry, North Massapequa. I still love you. But please, let's encourage the replanting of trees and the right trees for the right spot. Um, Taking the lessons home, we use ecological succession and competition theory to make the plantings look more purposeful. So one of the mistakes that I find that like a lot of native plant gardeners do is they treat every project as sort of like an ecological restoration project. And biodiversity is important. This is why I've dedicated my entire career to the enhancement of biodiversity and whatnot. But I'm sorry, like a New York aster that gets 9 feet tall and flops and doesn't bloom into the fall should not be in your front foundation garden. And so, we're looking at species that can pack this ecological punch and look purposeful at the same time. So we think about, and this is sort of relates to that ecological succession thing. We think about vertical layering and horizontal layering as you're going along. And if you introduce this competition in these communities, they prevent a lot of the flopping and a lot of the issues that unfortunately are giving native plant enthusiasts a little bit of a black eye, especially in the more built up western portions of Long Island. People are getting ticketed for native plant communities, and I think that part of it is thinking about gently moving Long Islanders in a direction. Have a wild backyard, your front yard, have native plants, develop an ecosystem, but be thoughtful about what you're planting and where and how it's going to grow together. It doesn't have to be an all or nothing proposition. I have kids. I have a bit of a lawn, I'll admit, but the kids spend more times on the margins of the lawn looking at bugs and finding stuff than they do on the lawn at any time. And so each year we continue to bite away at the margins and increase the native plant community in our yard. Our neighbors are amazed. They tell me that I've been here for 25 years. I've never seen a hummingbird since he moved here. There's fox in our yard and just random suburban Huntington, right? So we're excited about it, and it's honestly cathartic for me as a passionate environmentalist and a professional restoration ecologist. Is just how quickly nature can turn the corner if given a chance. And so we get a lot of doom and gloom out of these big multinational organizations. I donate to them too. So I get these letters in the mail saying that if you don't donate more, we're all going to die. But the people want to know what they can do in their yard. People want to know how to help, their space. And it's really cathartic for me to know that one little 100 square foot garden, especially if connected to your neighbors doing 100 square foot gardens, will make such a huge impact on so many different levels, on stormwater management, on climate change adaptation and mitigation, on pollinator connectivity. We have the capacity as humans, I believe, to turn this whole thing around. We just have to be thoughtful about doing it. And I'm so grateful for this organization and all of the like-minded individuals who are listening to me talk about the work that we're doing. So that's it. And I really appreciate the time you've taken and listening to my talk about fuzzy sets and ecology. So thanks. Yeah. Well, so when you have so nature is organized as such. Like you have a a beautiful oak forest. It's a really deep field, right? It's stable. It's resistant to perturbations. But if it was only that, you'd have a monolith. All we would have is deep, stable oak forest, right? And so nature's answer to that is adding a little bit of disturbance. So that's what, as topography changes and as water is increased to the beavers build dams, things flood, you introduce this diversity. And so what you're left with is this braided beautiful let's just say gradient between a healthy oak upland and a wetland that is a forested wetland that functions as a whole unit. Now, if you do dopey things that humans do, like concentrate lots of stormwater and put it to one spot, or rip out all the native species or so on, what you're left with, it just kind of drifts into this PC3 disturbance area. And so what happens is like, we see a lot of this in the context of what's referred to as old field, succession. So basically, a lot of New York State used to be farm fields and actively farmed. And so now, a lot of them are kind of left fallow and to see what kind of, what comes back up. And what happens is you start to see the early stages of ecological succession coming along. But what happens, the invasive species essentially lock it into early successional stages that are leaky from a nutrient standpoint. The oak trees can't get, established and so on. So the disturbance, this ecological drift, is basically flattening this complexity, this redundant, stable base of nature that we had. Typically, it's humans that are doing that. It's our manipulations of the landscapes that are doing it. The flip side of that, though, is that we could put it back, right? I think of nature as sort of like a Jenga game, right? Like when you push out the bricks, you keep pushing out the bricks. At some point, the whole thing collapses, right? But you could also rebuild the tower, put the bricks back, and so on. And I think that, on a small scale, and we're hoping to increase the scale, that's what we're doing here on Long Island. We're pulling it away from that anthropogenic disturbance and pushing it towards this more braided dynamic equilibrium that exists in nature and healthy portions of nature. Yeah, thanks. Anything else? Yeah, Marsh.
What stage we have
Yeah, I think about 250 to 500 plots, across Long Island. 500 would be a pretty monumental undertaking. 250 would be doable in a field season. Put them in a grid, take standardized data, run them through the workflow, and we have all of Long Island. Yes. Yeah. Carolyn? Those were clients' houses. Yeah. This is my kid though. Actually here pulling plants away on the cart from the Limpy sale. So that yeah, my house is like kind of crazy to be honest. Yeah, it's a little bit of a you don't know what a plant does until you live with it for a while. I've lived with a lot of different plants for a while in my yard, and my wife is very nice about experimenting. Yeah, good. Okay, thank you.